Chapter 23, we talked about how acetyl-CoA, formed from each of our major biomolecules, is metabolized for energy in the citric acid cycle and the electron transport chain. In Chapter 24, we talked about how carbohydrates are taken into the body and can be used to supply energy through central metabolism or through fermentation in anaerobic conditions. We also talked about how they can be used to store energy. In Chapter 25, we're going to be talking about how triacylglycerols get taken into the body, digested into acetyl-CoA for energy. We'll also talk about how acetyl-CoA can be used to make fatty acids, cholesterol, and a special type of molecule called ketone bodies. Because we've already learned about hormones, we'll also be paying attention to how the new routes we introduce are affected by the hormones glucagon, insulin, and epinephrine. Just like with carbohydrates, we want to start talking about lipids in the form that they enter our bodies as food. Most of the lipids in food are triacylglycerols, so we'll focus on those for simplicity. We'll talk about how they get absorbed into our bloodstream as fatty acids, and the remainder and majority of this chapter will be more closely related to fatty acids and other metabolites. All of this is set against the backdrop of the citric acid cycle and the electron transport chain, which are the only ways that we get energy from fatty acids. There is no anaerobic metabolism for fatty acids. Just like with carbohydrates, we start triacylglycerol digestion by eating triacylglycerols. Unlike with carbs, we don't do anything with them in the mouth. This has everything to do with the physical properties of lipids compared to the physical properties of carbohydrates. Lipids are, by definition, insoluble in water. They tend to stay in large globs of fat that aren't accessible to enzymes until they get into the stomach. In the stomach, the churning action of the stomach breaks up those globs into smaller chunks called chyme. You can see these chunks if you vomit. I know at your home, but don't try this at home. The chyme has more surface area, and some enzymes, gastric lipids, can start acting on the lipids. Gastric here just means stomach. It isn't the substrate for the enzyme in this case. Lipases hydrolyze lipids, but only partially here. This is important because it means that at least some of the surface of chyme has become more polar. In the intestines, additional lipases can continue to work on hydrolyzing lipids, with aid of biolipids to improve their solubility. With many fatty acids free, their polar ends can interact with water and make anything unhydrolyzed more soluble by forming a micelle. Micelle structures, which you can see on the top left, are similar to intestinal cell membrane structures. There's a polar outside and a nonpolar inside. The polar outside of the micelle can interact well with the polar outside of a cell membrane and essentially adhere or stick right onto the membrane. The micelle can then open up the membrane and flip all of its fatty acids into the membrane, into the outer membrane surface. They can then be flipped to the inside of the cell by enzymes called flipases. They can be pulled into the cytosol and then packed up inside of those intestinal cells by reassembling into triacylglycerols and then packing them inside of lipoproteins and membrane lipids, essentially creating a big soluble cell-like structure containing the triacylglycerols. These are called chylomicrons. These chylomicrons are exported from the intestinal cells into the lymph system. They are too big to be absorbed directly into the bloodstream. The lymph system distributes these packets into the bloodstream and hydrolyzes them once they, again, into the fatty acids and some mixture of mono and diglyceride micelles that can then move freely through the bloodstream and be absorbed by cells. From there, they can be used or stored. This summary of lipid digestion makes clear the many transitions that triacylglycerols need to go through to make it into the bloodstream. This is different than other biomolecules because triacylglycerols are insoluble, and fatty acids only limitedly soluble. The triacylglycerols need to be physically broken into smaller bits, hydrolyzed to more polar fatty acids and mono and diglycerides, in order to have enough surface area to be absorbed into the intestinal cells. Then they need to be packaged as those chylomicrons to be exported into the lymph system and then hydrolyzed again to stay soluble outside of the chylomicron package. It's a lot of work, but the energy density of fatty acids makes it well worth our while, which we'll see later on in this chapter. Again, we can either store lipid energy as triacylglycerols, or we can use it directly. In most cases, since we're taking in lipids at the same time as we're taking in carbohydrates, that means insulin is going to be active. Insulin, as a hormone, is primarily responsible for promoting the uptake and use of glucose, meaning that it will trigger glycolysis as a major form of energy production, but slow down the use of fatty acids for energy. So most of our fatty acids are stored initially and then released later. Fatty acids are the major way that energy is stored in our bodies as triacylglycerols. Notice that glycogen can store some energy and blood glucose even less. That energy tends to be used up fairly quickly, and it tends to be distributed largely through muscle tissues, whereas triacylglycerols store a far larger amount of energy and localize it in cells called adipocytes that exist primarily to store fat. These cells will make up what we call adipose tissue, that are tissues that are rich in fat. These fats will be stored until we need them. When the glucose levels drops, insulin also decreases and glucagon increases, triggering the use of lipids for energy whenever possible. Similarly, epinephrine, as all the energy I can get right now, I need it, like, please give it to me type of hormone, will trigger the use of lipids for energy while continuing to use glucose as well. The release of triacylglycerols by any hormone is accomplished by a hormone-dependent lipase. As soon as the hormone is active, the lipase is triggered to be active, and these lipases will hydrolyze triacylglycerols back to fatty acids that can re-enter the bloodstream to be used by any tissue that needs them. This takes a lot longer though, so fatty acids are not really a good source for quick bursts of energy and are much more commonly used for long-term energy. Triacylglycerols contain 4 parts. 3 of those parts are fatty acids, and those are all tied together on one glycerol. The fatty acids are lipids and get used for energy in a lipid-specific pathway. The glycerol is actually really closely related to carbohydrates and can enter the 3-carbon phase of glycolysis to give us some small amount of energy. The fatty acid parts need a special pathway to produce energy, completely independent of glycolysis. This pathway is called fatty acid degradation, or oxidation of fatty acids, or catabolism of fatty acids. Sometimes it's also referred to as beta oxidation, which really refers just to the last part of this process, a cyclic set of steps that gives us all of the energy out of this process. The entire process of oxidation has 3 steps. The first step is an activation step. ATP is hydrolyzed all the way to AMP, that's adenosine triphosphate going to monophosphate, to give us the energy to attach a fatty acid to coenzyme A. Notice 2 things about this step. The first is that ATP is degraded all the way to AMP, which is equivalent of spending 2 ATPs, not just 1. The other is that the acyl-CoA that is formed is essentially just a longer acetyl-CoA. Acetyl-CoA is just an acyl-CoA with specifically 2 carbons. The second step is transport. Transport brings the acyl-CoA into the mitochondria to be oxidized there. The acyl group is moved off of coenzyme A onto a molecule called carnitine that has a specific membrane transporter. Once inside, it moves back onto coenzyme A. And it's really important that the acyl-CoA gets oxidized in the mitochondria because that means that all of the cofactors produced by that oxidation can be readily reduced in the electron transport chain for as much energy as possible. This is different than how glycolysis produces an NADH that then has to be transported into the mitochondria later, which costs us some of the energy we would have otherwise gotten. The third and final step of the oxidation pathway repeats a series of steps over and over and over again until the acyl-CoA becomes shortened all the way to acetyl-CoA. This recurring set of steps is called beta oxidation. These first 3 of these steps follow a pattern that we've seen also in the citric acid cycle. First, an oxidation of an alkane to an alkene using FAD and yielding FADH2, then hydration to an alcohol, and then oxidation of an alcohol to a ketone using NAD+ and producing NADH. This pathway also produces the same cofactors that we saw in that portion of the citric acid cycle. FADH2 for the oxidation to an alkene, and NADH for the oxidation of an alcohol to the ketone. The fourth step of beta oxidation splits the two ketone molecules into two acyl-CoAs. One of the acyl-CoAs is specifically acetyl-CoA, 2 carbons, and the other is a carbon chain, an acyl group that is 2 carbons shorter than it was before. At the end of this step, we have an acyl-CoA 2 shorter than we started, an NADH worth 2.5 ATP, an FADH2 worth 1.5 ATP, and an acetyl-CoA that's worth 10 ATPs. So for every 2 carbons we shorten a fatty acid by, we gain a total of 2.5 plus 1.5 plus 10 ATP, or 14 ATP. This pathway is only oxidative. There's no anaerobic pathway for fatty acids. It's helpful to think about the 3 steps of fatty acid oxidation, the activation, the transport, and then beta oxidation, in terms of how we'd use that to calculate the yields for an entire fatty acid. No matter what, our fatty acid will always go through an activation that will always cost us 2 ATP. The energy gain will be mostly through beta oxidation, the 3rd step, so we need to figure out how many times that beta oxidation occurs. Here we show stearic acid, which has 18 carbons. The first beta oxidation gives us an FADH2, an NADH, an acetyl-CoA, and a 16-carbon acyl-CoA. So we've gone from 18 to 16. The next time we go through beta oxidation, we have FADH2 produced, NADH, acetyl-CoA, and now a 14-carbon acyl-CoA. So again, going down by 2 every time. This repeats 8 times. After the 8th beta oxidation, the 2 acyl-CoAs that are produced are both acetyl-CoA. When we go from 4 down to 2, it splits into 2 acetyl-CoAs because a 2-carbon acyl-CoA is acetyl-CoA. So we don't need to go through beta oxidation for those very last 2 carbons. They just enter the citric acid cycle. So the overall yield of ATP for stearic acid is 8 times 1.5 ATP for the FADH2s produced in those 8 beta oxidations, plus 8 times 2.5 ATPs for the NADHs produced in 8 beta oxidations, and 8 times 10 ATP for the 10 acetyl-CoAs produced in 8 beta oxidations. If you're looking at the calculation on the bottom, notice there's also a plus 10 ATP. That plus 10 is for the acetyl-CoA that doesn't need to go through beta oxidation because it's already acetyl-CoA. And then the minus 2 ATP at the very end is for that activation step. If you add all this together, we get 120 ATP for one 18-carbon stearic acid molecule. We can calculate this total energy yield for any saturated fatty acid as long as we can figure out how many beta oxidations occur. Since each beta oxidation consumes 2 carbons, we take the total number of carbons, in this case 18, and divide by 2. For stearic acid, this gives us 9. 18 divided by 2 is 9. 9 2-carbon pieces. However, we don't do beta oxidation on the last 2-carbon piece. It's always already acetyl-CoA. So we have to subtract 1. So we take 18 divided by 2, that's 9 2-carbon pieces, but we subtract 1 from the number of beta oxidations we actually go through. So this gives us 8 beta oxidations. 8 beta oxidations, which are each worth 14 ATP apiece, plus 1 extra acetyl-CoA worth 10 ATP, minus 2 ATP for the activation step, gives us a total of 120 ATP. So a different way of looking at that same calculation. This is worth comparing on a per carbon basis to glucose. Stearic acid gives us 40 ATP, 120 divided by 3, for every 6 carbons, while glucose only gives us 30 ATP. Longer molecules will give us even more energy per carbon. So fatty acids store energy much more efficiently than carbohydrates. We can do all the same calculations we just did for an unsaturated acid, except that every time there's unsaturation, we need to subtract an FADH2. This is because if we already have a double bond, we can't get the FADH2 associated from its production. We often need to move the double bond, which we can see in the second and third images on this slide, but that doesn't really change the energy yield at all. So for example, if we had 18:2, a fatty acid with 18 carbons and 2 double bonds, we'd get the same energy we would for 18:0, which we just calculated as 120 ATP, minus the energy from 2 FADH2s. In other words, we'd get 120 ATP, that's from the 18 carbons, minus 2 times 1.5 ATP, that's from the 2 FADH2s that we do not actually produce because of the unsaturation. This calculates to 117 ATP from 18:2. So we know that triacylglycerols can store energy much more densely than glucose, even when they're unsaturated, which makes me, and maybe a few of you, wonder why we have glucose at all. Fatty acids clearly store energy more densely per carbon, and even more densely when you take into consideration that the triacylglycerols don't need to be surrounded with water in the way that carbohydrates do. However, fatty acids are really difficult to mobilize. They're stored in specialized tissues, separate from where often our energy need is, and they need to be activated to release the fatty acids. And then they need to get to the right tissues. They make really good energy supply for functions that are always on, or for long-term use, but they make terrible surge energy. So skeletal muscles might use fatty acids to supply resting energy, they do, but they will switch to glycogen anytime they are being active. Similar, cardiac, that's heart muscles, can use fats most of the time, since your heart should always be on. But when it starts to work harder, it can use other energy sources as well. The liver, on the other hand, always prefers fatty acids and can use fat-based energy to help support glucose-based functions elsewhere, most notably in the Cori cycle that we saw in the last chapter. The brain poses the most interesting application of fatty acids. Your brain should be always on, but your fatty acids can't cross the blood-brain barrier, which prevents their use to supply brain energy. Your brain prefers glucose, but can also use a special metabolite of fats called ketone bodies to deliver energy from fats when glucose is limited. Since fats are unable to supply energy for the brain, but are likely to be the main energy source during periods of fasting, it's important to have some way of getting fatty acid-based energy into a form that can pass through the blood-brain barrier. We can't make glucose from fatty acids because there's no route from acetyl-CoA back up to pyruvate. Instead, we make a class of compounds called ketone bodies. There are 3 ketone bodies, all shown in the top of this slide: acetoacetate, beta-hydroxybutyrate, and acetone. They're all initially produced as acetoacetate, the leftmost molecule. Beta-hydroxybutyrate in the center is just a reduced form. Notice the change from a ketone to an alcohol. This is formed with the aid of NADH, which is shown on the bottom left. The rightmost molecule, acetone, is a decarboxylated version of acetoacetate. That means carbon dioxide has been removed from it. Acetone isn't actually a really good source of energy. You can use it a little bit in the liver, but instead helps to balance drops in pH that are associated with ketone body use. The process of making ketone bodies is called ketogenesis. Based on the name, is the process anabolic or catabolic? There are several steps to make acetoacetate, which are essentially just joining acetyl-CoA pieces together. 2 acetyl-CoAs are added together initially, which you can see in the first row. A third acetyl-CoA needs to be added to actually separate out the acetoacetate, which you can see in the second row. However, it gets recovered, and which you can see in the third row. The acetoacetate then is worth 2 acetyl-CoAs, or about 20 ATP. Acetoacetate can also be reduced to beta-hydroxybutyrate after it's formed, which is another ketone body. Beta-hydroxybutyrate can be oxidized back to acetoacetate and form NADH, so it's essentially storing up energy from the NADH in addition to the 2 acetyl-CoAs. It's essentially worth 22.5 ATPs, 20 from the 2 acetyl-CoAs and 2.5 from the NADH. This slide doesn't show us the actual molecules, but it helps to get a general way to think about how the ketone bodies are formed, essentially storing the energy from 2-acetyl-CoAs and, in the case of beta-hydroxybutyrate, also storing the energy of an NADH. When we need to use acetoacetate, or beta-hydroxybutyrate, we need to get it back onto a coenzyme A molecule before we can split it up into the 2-acetyl-CoAs. In order to do that, it swaps places with a succinyl-CoA, which is already on coenzyme A. Succinyl-CoA is produced in the citric acid cycle, but normally when it's produced there, that CoA is hydrolyzed in order to phosphorylate a GDP and produce a GTP. It can't do that when it's used to process the ketone bodies, so we lose the equivalent of about 1 ATP when we're metabolizing ketone bodies. It's a small price to pay to feed our brains, though. The real problem with ketone bodies isn't the really small loss of energy. That's a small problem. A much larger problem is that acetoacetate and beta-hydroxybutyrate are the conjugate bases of acids, and when they're produced, they are going to drop the blood pH because they are acids. When the ketone bodies are present at elevated levels in the bloodstream, it's called ketosis. A slight case of ketosis will drop the blood pH slightly. A severe case drops the blood pH severely. Recall that with changes in pH, enzymes are no longer likely to fold as well and will drop their activity rate as they move away from the optimum pH. So whenever we produce ketone bodies in large quantities, this has a cascade of other effects on the bodies. One of these effects is that hemoglobin doesn't carry oxygen as well as it once did. In mild situations, this can be solved by eliminating some excess acidity by converting acetoacetate to acetone and carbon dioxide, which you can see on the bottom left. Both of the acetone and the carbon dioxide can be excreted through our lungs. Acetone can also be processed for a small amount of energy in the liver as well. That whole process is normal and happens to some extent when we're not eating for a while, like when we're sleeping. It's not a big deal. You can actually smell the acetone on people's breath if they're going through diet changes sometimes. And I can smell it on my kids' breath every morning before they eat breakfast. It's endearing, except, when it means they're hangry too. In order to be used for long-term energy, as we described in the first part of this chapter, we also need to be able to make fatty acids and lipids to store energy or to fill other roles. The remainder of this chapter will be talking about how acetyl-CoA can be used to build up fatty acids, a process called lipogenesis, and also how acetyl-CoA can be used to build up cholesterol, and then thinking about how all these pathways, both the catabolic and anabolic, fit together with our hormonal control. Lipogenesis, which again is the anabolic pathway where we synthesize fatty acids from acetyl-CoA, is very similar to the reverse catabolic counterpart, fatty acid degradation. Because we sometimes need to synthesize different fatty acids than are available, both processes will sometimes occur at the same time. For this reason, they need to be separated and controlled individually. To accomplish this, our bodies will perform lipogenesis in the cytosol, physically separating it from the fatty acid degradation that happens in the mitochondria. It also uses different enzymes that can be controlled separately, and a different carrier molecule for the fatty acid and the carbon source. We also spend energy in a very particular way. NADPH is the main source of energy as we build up a fatty acid. All these differences are important to maintain independent control over these mostly opposite pathways. Because we usually generate acetyl-CoA in the mitochondria, whether it's produced from pyruvate or from fatty acids, the first thing that we need to do to synthesize new fatty acids is to move it out into the cytosol. This is the purpose of the citrate-malate pathway. Citrate and malate can both move in and out of the mitochondria fairly easily with specific transporters. They're also both components of the citric acid cycle. Inside the mitochondria, we essentially run steps 8 and 1 of the citric acid cycle to convert malate to citrate. Notice that it produces an NADH in this process and takes an acetyl-CoA and moves it onto oxaloacetate to make citrate. The citrate can move into the cytosol, which we see on the bottom of the figure, and in the cytosol, we'll spin an ATP to move the acetyl-CoA back off of citrate and make oxaloacetate, essentially running step 1 of the citric acid cycle in reverse. Then we spin an additional NADH to run step 8 in reverse. While it seems like we might be losing energy, this actually gives us a way to move NADH electrons in the cytosol from glycolysis into the mitochondria, so it's essentially a wash. Acetyl-CoA is the initial source of all the carbons in lipogenesis, but we need to move them onto a carrier protein that will be able to be recognized by the lipogenesis enzymes. You can see this step in the top row, essentially just swapping out the acetyl group from a CoA onto the acyl carrier protein called ACP, which literally is an abbreviation for acyl carrier protein. This is just for the first two carbons. Everything else needs to be added to this in two carbon sections, kind of the opposite of beta oxidation. We also need to activate the two carbon piece we're going to add on, because it's harder to add carbons together than it is to break them apart. We do this with an acetyl-CoA by capturing a carbon dioxide, which costs an ATP. You can see this in the second row. This carbon source also needs to be moved onto an acyl carrier protein so that lipogenesis enzymes can recognize it as well. So the malonyl that's produced on a CoA gets moved onto an acyl carrier protein also. This resulting malonyl ACP, which we can see on the bottom, is the source of every two carbon addition to the chain after the first and represents an investment of an acetyl-CoA and also the one ATP used to activate it. Starting from an acyl group and increasing by two carbons is a process that happens repeatedly in lipogenesis, just like beta oxidation repeated over and over again in fatty acid degradation. The first step, which you can see on the top of the left side, joins the main acyl group with the activated carbon source, malonyl ACP. Remember from the last slide that malonyl ACP was already worth an acetyl-CoA and an ATP, essentially 11 ATP altogether. The resulting 1,3 ketone, the 1,3 just referring to the relative positioning, goes through the opposite process that we saw in beta oxidation. In this case, a reduction to an alcohol with NADPH, dehydration to an alkene, and then reduction of the alkene to an alkane, in this case, again using NADPH. At the end of this process, we have an acyl group that's 2 longer than it was before, and we spent the malonyl ACP worth 11 ATPs, and also 2 NADPHs, which are the equivalent of 5 ATP. Each cycle of lipogenesis is then worth 11 plus 5, or 16 ATP. Note that this is more than the energy we get out of beta oxidation, which was only 14 ATP. Just like with glycolysis versus gluconeogenesis, the anabolic pathway costs more than the catabolic pathway generates. Nothing is free in life. Again, this set of 4 steps will repeat for every 2 carbons that are added until the chain reaches the desired length. The first 2 carbons don't need to be activated or reduced, so they just cost 1 acetyl-CoA. But every set of 2 additional carbons costs an acetyl-CoA, an ATP to activate it, and 2 NADPHs for the reduction, an equivalent of 16 ATPs. So for the 16 carbon fatty acid shown, we'll have a number of cycles equal to 16 carbons divided by 2, since the carbons are added 2 at a time, minus 1, since the acetyl ACP isn't cycled, it's just transferred from acetyl-CoA. 16 divided by 2 minus 1 equals 7 cycles. To figure out how much energy was spent to make this molecule, we'll say that 7 cycles times 16 ATP per cycle plus another 10 ATP for the acetyl ACP that isn't part of a cycle. So 7 times 16 plus 10 is 122 ATP in order to produce a 16 carbon fatty acid. We can't make unsaturation at all locations on a fatty acid, but we can make some unsaturation in our bodies. We accomplish this by using oxygen to oxidize both an alkane to an alkene and NADPH to NADP+. This means that it actually costs us energy to oxidize an alkene, where in all other oxidations we gained energy. We consider NADPH essentially like NADH, so again it's worth 2.5 ATP. Each double bond then costs us an additional 2.5 ATP to make. So when we calculated the yield of 16 carbon fatty acid last slide, we found it cost 122 ATP to make. If we had a 16 carbon fatty acid that also had one double bond, 16:1, it would cost 122 plus 2.5 ATP, or 124.5 ATP. If we had a 16 carbon fatty acid that also had two double bonds, it would cost 122 plus 5 ATP, or 127 ATP. Acetyl-CoA isn't just used for synthesizing fatty acids. It can also be used to make cholesterol. Your body not only makes the cholesterol, but it also makes the lipoproteins that carry it, either a high-density lipoprotein, HDL, or a low-density lipoprotein, LDL. The majority of your cholesterol comes from your own biosynthesis, not actually from your diet. And even when it does come from your diet, in both cases, your body decides which carrier to give it. So ingesting cholesterol really isn't what gives us cholesterol problems. High LDL, that's again the low-density lipoprotein that transfers cholesterol out into the tissues, that has a lot more to do with eating trans fat. Remember, trans fat usually comes from hydrogenated or partially hydrogenated oils, whether or not it's shown up on a nutrition fact label. Smoking can also lower the HDL levels. That's generally the good cholesterol. So, don't. Regular exercise can improve HDL levels, particularly if people hit certain thresholds of activity, typically starting at about an hour of moderate intensity exercise a week. You might not realize this, but Diplodocuses also metabolized fatty acids. And to tell you a little bit more about that, I have a younger Professor Boscher, Professor Levi, to explain.
Diplodocus name means double bean.
Is there anything else you wanted to tell them about?
And it lived during the Jurassic, and it aged, and it didn't survive into the late Cretaceous, and what else could I tell them?
Maybe what type of dinosaur it is.
A sauropod?
Yeah, thanks, Professor Levi. It's always helpful to see pathways all together to see how they're connected. Fatty acid degradation and glycolysis are both routes to supply energy aerobically. Fat is a much better way of storing energy, and our bodies can store a lot more of it than glucose or glycogen, so we're always going to use glucose preferentially first. With this in mind, we're going to run through a few scenarios of what might be happening in our bodies. The first is that we have just eaten a big meal. If we have just eaten, we will start to take up monosaccharides, fatty acids, and amino acids in pretty large amounts. That means that glucose levels are going to surge in our bodies, and our bodies will release insulin. Insulin will promote using up all that extra glucose. In order to do that, we'll increase the rate of glycolysis to the point where it's supplying the majority of our ATP. And since we only need so much ATP, we're also going to decrease fatty acid degradation at the same time. This means that most of the fats will be stored for later in adipose tissues, so fatty acid catabolism will be fairly low. If we still have really high glucose as we meet all of our ATP needs, which is actually pretty likely, our bodies will also make glycogen to store for later, and we'll also use the pentose phosphate pathway to power lipogenesis as well. Ketone bodies won't be produced since there's plenty of glucose. Gluconeogenesis will also not be active unless exercise is occurring at the same time. Okay, for a second scenario, let's say someone is sleeping. They haven't eaten for a while, so their blood glucose level is going to drop. That triggers the release of the hormone glucagon. Glycogenolysis may occur to supply some additional glucose, and the majority of ATP production is going to shift away from glycolysis to fats and be produced through fatty acid degradation. Because these can't supply the brain with energy, they will also be used to perform, the acetyl-CoAs from fatty acid degradation will also be used to perform ketogenesis and increase the ketone body concentration. That means somebody who is sleeping will have a decreased blood pH to some extent. Very little lipogenesis is likely to occur since there isn't a lot of extra energy. Gluconeogenesis also probably won't occur either since we can't make glucose from acetyl-CoA and there's no new source of pyruvate or lactate in order for us to synthesize glucose from. Okay, so now our third scenario is someone who's on a keto diet, where they primarily consume fats while also limiting carbohydrates and protein intake. Since glucose levels will remain low, glucagon remains the dominant hormone, and most of the ATP produced will come from fatty acid degradation. This will look a lot like fasting in the last scenario. Depending on how long the carbohydrates stay low, though, and how low they go, this can be pretty dangerous. Ketosis will decrease the blood pH and makes it a lot harder to breathe. The longer you're supplying glucose-like energy through the ketone bodies, and the higher those ketone body levels get, the more problems one is likely to see. In the next chapter, we're also going to talk about how the metabolism of amino acids from proteins fits into this larger metabolic picture as well.